The Kachemak Bay National Estuarine Research Reserve is leading an effort to better understand the application of environmental DNA, or eDNA, through a collaborative approach with partners within the University of Alaska system and partners at the NERR. eDNA is a genetic material of species that is found in an aquatic environment or terrestrial environment. This method of detecting biota is a passive and non-destructive way to conduct biological research. This project is threefold, including an Alaska-based researcher-to-researcher workshop exploring the capacity for eDNA analyses and a community workshop focusing on knowledge sharing. eDNA gets at the most basic ecological question of who is there within an ecosystem. In Kachemak Bay, this question can be extremely difficult to answer. Vast and remote coastline translates into a challenging region to track larger ecosystem changes, and eDNA is proving to be a useful tool in filling many of the gaps of species monitoring, particularly in this high-latitude, remote environment. The target users for this project are coastal decision makers of the northern Gulf of Alaska region, where monitoring ecosystems is difficult due to the remote nature of the region. This project builds on previous NERRS collaborative projects, such as Estuaries DNA and community-focused workshops from the NERR. This project addresses two management needs of the NERR. First, to apply novel techniques for environmental monitoring, remote sensing, and community monitoring in order to expand information and skill sets available to coastal decision makers, and second, to analyze and outreach ecological data sets in the context of coastal management priorities for stakeholders in subarctic ecosystems.
As multiple users continue to collect data for disparate eDNA projects in Alaska, we propose a collaborative effort to share techniques for various questions and connect those users that are currently investigating similar projects. The timing is right for this project, as an ad hoc group of scientists working with eDNA in Alaska has formed over the last year, coordinated by researchers at the University of Alaska Fairbanks. This monthly meeting has been a great way to connect to the research community and jumpstart the first collaborative component, researcher-to-researcher networking. Projects shared have generally addressed different aspects of ecosystem resilience in the face of rapid environmental change, providing novel processes to identify and monitor current species communities. Additionally, we use the reserve platform for engagement, including Kachemak Bay and Lower Cook Inlet Marine Ecosystem Work Group, the Kachemak Bay Science Conference, and informal lunch publication clubs to engage our local community of coastal professionals around this work. Kachemak Bay Research Reserve operates with the goal that research in our region should reflect community input on species of cultural, economic, or ecological importance. To move towards that goal centered on eDNA projects, we are hosting a workshop based on our cross-reserve collaboration between our partners at the NERR, who have established a framework for eDNA knowledge sharing, and our regional partners here in Kachemak Bay.
This project brings together researchers and decision makers from around the state of Alaska to discuss the uses of eDNA, sharing knowledge and experiences that can help push these methods forward. While Alaska is a large state, the research community is relatively small and relies heavily on each other for resources. These workshops address common hurdles and detection methods for eDNA, while also strengthening relationships and creating new partnerships between researchers.
The community workshop in Kachemak Bay will be an opportunity to share eDNA methods and information pertaining to aquatic eDNA projects throughout Alaska and discuss community interests with the goal of understanding how scientists and communities can utilize eDNA to answer research questions in Kachemak Bay. Participants will discuss successful approaches and applications at local and regional capacity to maximize the power of this ecological tool, identify emerging needs and opportunities, and form new partnerships. This workshop includes an activity to demonstrate the concept of eDNA, a panel of scientists putting eDNA into practice, and hands-on and field-based demonstrations of eDNA sampling and initial processing. This learning is intended to inspire resource and land stewards and managers to brainstorm applications for their places within other communities and across other natural environments in coastal Alaska. The team will model this engagement after the successful hands-on workshop held with co-management partners at the NERR in Hawaii in 2022. The NERR has set a precedent for how to communicate complex technology to a diverse audience while engaging community members, subsequently providing a template for the NERR projects team.
Hi all, this is Debbie Bogie Tobin from the University of Alaska, Anchorage, Kenai Peninsula College, Kachemak Bay campus. We use eDNA in the classroom to engage with our students in a variety of ways. First off, I have been sampling the Kenai River looking for sources of prey and beluga whale signatures for the last several years in conjunction with NOAA and National Marine Fisheries Services. And so each month the students go with me during the academic year to collect eDNA samples, and then we filter those samples back in the lab on campus. So it gives the students some real life valuable field experience and tools of the trade experience. I also use eDNA with a current harbor porpoise permit project that I have in conjunction with NERR. And so part of my workload involves identifying hotspots for harbor porpoises in Kachemak Bay and Lower Cook Inlet. And this summer we will be beginning to use eDNA as well and engaging both graduate students and undergraduate students who have rolled over from the previous Semester by the Bay courses with me. So we'll be collecting samples to see similarly what is available for prey for the harbor porpoises in parts of Kachemak Bay, and also looking for some signatures of the harbor porpoises and other marine mammals themselves.
This project will support development of local protocols for eDNA collection and analyses, and an in-depth inventory of current eDNA projects in the Kachemak Bay region. This will lead to the cultivation of shared research questions for researchers working in the northern Gulf of Alaska, and strengthened relationships among such entities as academic, state, and federal agency staff, government staff, tribal entities, and non-governmental organizations will be a product of this project. And this is currently building community among research and teaching components within the University of Alaska system in particular. With help from partners, the Kachemak Bay National Estuarine Research Reserve intends to integrate eDNA sample collection into current research and monitoring efforts. By the end of this project, the greater Northern Gulf of Alaska region community will be set up to use eDNA in-house to co-develop research and monitoring questions.